Assalamualaikum and good morning. My name is Izzah binti Bahari and today I will be presenting my practical training 2 on pest and vector control. In this presentation, I will share the activities, knowledge and practical exposure that I gained throughout my training. So let's begin. Pest and control is an important component of environmental health because pests and disease vectors can contribute to the transmission of various diseases. During my practical training, I was exposed to several activities involving vector surveillance, pest control, environmental management, and disease prevention. These activities included mosquito surveillance and control, rodent surveillance and control, as well as other field-based activities carried out to reduce the risk of vector-borne and zoonotic diseases. The following slide will highlight the activities that I observed and participated in during my practical training. The first major area of my practical exposure was dengue control. Dengue control focuses on the surveillance and control of Aedes mosquito populations to reduce the risk of dengue transmission. During my practical training, I was exposed to several activities, including Aedes larva surveys, larva identification and preservation, insecticide preparation, and chemical dose calculation, as well as thermal fogging. I was also exposed to the interpretation of Aedes larva survey findings to understand potential breeding risks within the inspected area. These activities provide me with practical exposure to how dengue vector surveillance and control measures are implemented in the field. As part of the dengue control program, I participated in a place of breeding or PTP inspection at Apartment Teratai under the guidance of PPKP Faris and PKA Khairul Amin. The inspection focused on identifying potential Aedes breeding sites, particularly water holding containers and areas where stagnant water could accumulate. Suspected larvae were collected for further examination and identification. I was also exposed to the proper handling and preservation of the collected specimens. This activity helped me understand the importance of systematic inspection in detecting potential Aedes breeding sites and preventing mosquito population development. Under the guidance of the vector control personnel, I was exposed to the examination of the specimen using a microscope. Proper handling was important to ensure that the specimens were not damaged during the identification process. The identified specimens were then preserved in suitable containers, labeled as recorded in the larva identification record book. Before participating in the field fogging activity, I received practical exposure to insecticide preparation at the office. The vector control personnel explained the different type of insecticide used for vector control and demonstrated the correct procedure for measuring and preparing the chemical mixture. One of the insecticides introduced to me was ethylene. The preparation demonstrated during the training involved 160 milliliters of ethylene mixed with 4 liters of water. I also learned the importance of using appropriate personal protective equipment when handling insecticide, as well as the preparation and cleaning of the equipment used for fogging. After learning about insecticide preparation and equipment handling at the office, I was given exposure to the actual thermal fogging operation in the field. The fogging activity was carried out at Taman Benam Indah as part of dengue vector control activities. During the operation, I observed the preparation and operation of the thermal fogging machine by the vector control personnel. I also participated in the field activity together with the team. The activity provided me with practical exposure to the application of chemical control measures in the field and helped me understand the importance of proper procedures and safety precautions during fogging operation. Next, I would like to present the findings from the Edith Lava survey conducted at Jalan 1A, Taman Bernam Indah. A total of 40 premises were visited and 12 premises were inspected for potential mosquito breeding sites. Among these, 6 were identified as vacant houses. One positive breeding site was detected in a water holding bucket. The larvae collected from the site were examined and identified as Aedes aegypti. This finding demonstrates the importance of conducting thorough inspection because even a simple water holding container such as a bucket can provide a suitable breeding site for Aedes mosquito. After the dengue control activities, I was exposed to malaria control activities. During this exposure, PPKP Hisham provided a briefing on a recent malaria case, including the chronology of the case and the control activities that were carried out. I also learned about the importance of entomological surveillance in malaria control, particularly the surveillance of Anopheles mosquitoes and their potential breeding habitats. The surrounding areas of the affected location were assessed to identify suitable mosquito breeding habitats and to support the assessment of malaria transmission risk. This exposure helped me understand that malaria control involves not only case investigation but also entomological surveillance and appropriate vector control measures. Next, I was exposed to malaria control activities related to a malaria cluster at Sungai Seladang Wild Campsite in Hulu Benam. The control activities cover three main components. The first was source reduction, which involved physical, biological, and chemical measures to reduce potential mosquito breeding habitats. Larva source management was also introduced as part of managing potential breeding areas. The second component was the Anopheles larval survey. The surrounding environment, particularly areas near streams and forested areas, was assessed for potential Anopheles breeding habitats. I was also introduced to malaria vector species, including Anopheles introlatus and Anopheles maculatus. The third component was transmission control. The measures include indoor residual spraying, contact screening, blood slide collection, insecticide-treated nets, and continuous surveillance and follow-up. Next, I was exposed to filariasis control activities. Filariasis control requires an integrated approach involving mass drug administration, vector control, environmental management, surveillance, and health education. Mass drug administration or MDA is used to provide anti-filarial medicines to eligible populations and help reduce the level of microfilaria in the blood. Vector control focuses on identifying and eliminating mosquito breeding sites, while environmental management includes improving drainage, removing stagnant water, and maintaining proper sanitation. Personal protection such as using mosquito nets, repellents, and protective clothing also help reduce exposure to mosquito bites. Overall, these combined measures are important to interrupt transmission and reduce the long-term impact of filariasis. Next, I was exposed to fly surveillance activities conducted at Taman Iron Garden, Belian, and Famira Chicken Farm. The main purpose of the survey was to assess the density and distribution of flies at selected locations. The survey focused mainly on Musca domestica or the housefly. Activities include fly observation and collection, species identification, and recording of fly numbers to determine the fly index. Based on the survey findings, fly density at the assessed location ranged from high to very high, particularly at the Famira chicken farm. The presence of Musca domestica and the high fly index indicated considerable fly activity. Therefore, proper environmental sanitation, waste management, and continuous fly control are important to reduce fly breeding and infestation. Next, I was exposed to a study on three important mosquito genera, namely Anopheles, Culex, and Mansonia. Mosquito surveillance is important because different mosquito species have different breeding habitats, behaviors, and public health significance. Anopheles is particularly important because some species are associated with malaria transmission. Culex mosquitoes are commonly found in stagnant and organically polluted water, while Mansonia mosquitoes are closely associated with aquatic vegetation. Next, I was exposed to rodent surveillance and control activities at Ma'had Al-Mustafa, Ulu Tamu, Batangkali. The activity began with an assessment of rodent presence and environmental conditions that could support rodent activity. Several indicators, including rodent droppings, improperly managed waste, and potential accessible food source were identified during the inspection. Following the assessment, 40 rodent traps were installed at selected locations. 36 traps were placed around the Ma'had premise and 4 traps were placed near the river. Different types of bait, including salted fish and banana, were used to attract rodents. The traps were subsequently inspected and 3 rodents were successfully captured. The rodents were identified as Rattus rattus, consisting of 1 male and 2 female. The captured rodents were processed and their organs were collected for laboratory examination. The samples were then sent to the Institute for Medical Research or IMR for leptospira detection. The lab results show that Leptospira was not detected in all 3 captured rodents. The rodent index obtained was 1, which was classified as low. Although the rodent index was low, continuous monitoring, proper waste management, and environmental sanitation are still important to prevent an increase in rodent population and reduce the potential risk of leptospirosis transmission. The next component of my practical exposure was the vector quality assurance program. Quality assurance is important to ensure that vector surveillance and control activities are carried out systematically and effective. Throughout my practical exposure, I observed that accurate surveillance, proper documentation, correct control procedures, and continuous monitoring are important components of quality assurance. The final area covered under pest and vector control is pest and vector control at international entry points. International entry points are important from a public health perspective because the movement of people, goods, and transportation may provide opportunity for pest and disease vectors to be introduced and spread between areas. Therefore, control activities may include vector surveillance, pest control, environmental management, and regular inspection of relevant areas. In conclusion, pest and vector control plays an important role in environmental health and disease prevention. The practical training provided me with valuable knowledge and exposure to various vector control activities. This experience has strengthened my understanding of the importance of surveillance, proper control measures, and continuous monitoring in protecting public health. Thank you for your attention.